Welcome everyone to our October webinar, during which we'll be celebrating Gerald Gleich's legacy in eosinophil research with a very nice program, ending with a talk that will be given by the first recipient ever of the International Eosinophil Society Lifetime Achievement Award. First of all, I'd like to say a few words about our society. The International Eosinophil Society is an organization of scientists and clinicians who are interested in the eosinophil, a cell I'm sure many of you are well aware of, and a society which really sponsors a series of webinars like this one, but also holds a meeting every 2 years, which allows more networking amongst members and trainees that are involved in eosinophil research. Next slide. If you're interested in becoming a part of our society, I encourage you to visit our website, eventually also to scan this QR code. And please note that free membership is offered for early career individuals. So there's really no reason not to join us. We'd also like to underline that there'll soon be an opportunity to be joint members of the International Eosinophil Society and the European Mast Cell and Basophil Research Network. We've also very recently refurbished our website, and I encourage you to visit it. You'll see that it's changed significantly. It's become much more modern, user-friendly, and there are many resources that have been tailored to support our growing community. You can also scan the QR code or follow the link which is shown here. And we also have a special page dedicated to our upcoming Congress, which will be held in France next year during the summer. We also are highlighting on this new website journal reviews, which are submitted generally by individuals that are at the beginning of their scientific career in eosinophil research. And we encourage you also to visit these pages and eventually submit your own personal review that should be clear, compelling, and appeal to our membership. This is just an announcement of our upcoming meeting that I just mentioned that will be held in Montpellier, which is a beautiful city in the south of France, not far from the Mediterranean coast. The meeting will be held next summer from the 7th to 11th of July. And with this final introductory slide, I'd like to extend warm thanks to the members of our Corporate Advisory Council on behalf of our society. Before introducing the speakers, I'd like to remind you of a few technical issues. First of all attendees are muted and the chat feature is disabled. However, you can enter all your questions in the question and answer section, and these questions will be put to the speakers by the moderator. This webinar is being recorded, and the recording will be posted on our website after the meeting, after the webinar. Next slide. I'd first like to say a few words about the award itself, and then of course about Dr. Gleich. The International Eosinophil Society Lifetime Achievement Award is a distinguished honor that celebrates exceptional contributions to eosinophil research and a lifelong commitment to advancing the understanding and treatment of eosinophil-associated diseases. The 2024 award is being bestowed upon Dr. Gerald Gleich today, and he will receive the actual award during our Congress next summer. All those interested in eosinophils are familiar with the work of our first awardee. It is an enormous honor and pleasure for me to introduce Jerry, who is clearly the first and most long-standing eosinophilomaniac ever. His relentless work on eosinophil granule proteins has advanced our understanding of eosinophil involvement in tissue damage and dysfunction in many disorders. Jerry and his co-workers have developed means of detecting these proteins in tissue and body fluids, making it possible to decipher the role played by eosinophils even when they are no longer recognizable as such, because they have degranulated or undergone cytolysis. By the way, I encourage you to read the beautifully illustrated article he recently authored with his partner in life and work, Chris Leiferman, about the secret life of dead eosinophils. This slide gives a very brief overview of Dr. Gleich's outstanding career, starting as a medical student at the University of Michigan, and then serving many years at the Mayo Clinic, where he established a research lab for allergic diseases and chaired the Department of Immunology, among other distinguished functions. His most recent and ongoing engagement has been as professor of dermatology and medicine at University of Utah, where he continues to share his precious knowledge and know-how with yet another generation of lucky trainees and colleagues, and surely continues to ask those compelling questions he is well known for. Dr. Gleich has served on many prestigious committees and has received many honors and awards that would take way too many slides to enumerate. He has authored more than 700 articles. In short, Jerry is a pioneer. He is creative, curious, and enthusiastic about the future of eosinophil research, which is precisely the title of the lecture he will be giving at the end of today's webinar. Before that, we have several other fabulous speakers and topics. You will be hearing 4 presentations in all, and here's today's speaker lineup. As you can see, we have chosen a fine mix of physicians and scientists working on eosinophil biology, which is what our society is all about. You can find a detailed professional biography for each of these speakers on our website. Our first speaker, Stephen Ackerman, is professor of biochemistry and molecular genetics at the University of Illinois in Chicago, and a long-standing and very committed member of our society. Steve trained as a postdoctoral fellow with Jerry at the Mayo Clinic and has been exploring eosinophil development and functions in health and disease since then. His most recent contributions in terms of translational research include development of the eosinophil string test, allowing non-invasive monitoring of disease activity in eosinophilic esophagitis, and the development of a novel peptide nanoparticle CCR3 antagonist that interferes with eosinophil recruitment in tissues. Steve, we are impatient to hear all you have to say about Jerry's work and contributions to our current understanding of eosinophil functions and the roles they play in disease.
All right, thank you. Let me start sharing. Good morning, everybody. It's my pleasure and honor to provide the first presentation in this webinar to talk about Jerry's impact on the field and eosinophils and disease. Jerry has made huge contributions in terms of basic translational and clinical research, much of which is paradigm shifting. But beyond that, he is an amazing mentor. I spent 5.5 years as a postdoctoral fellow in his laboratory. He also has provided considerable service and leadership to the eosinophil field. And most recently, he's become entrepreneurial. So he's a Renaissance man in that regard. So let me just test you quickly and place Jerry in the context of luminaries in the eosinophil field. Who named but did not discover eosinophils? Who discovered them but did not name them? Who worked on eosinophil development, molecular biology, and remodeling? Eosinophils are unequivocal liars. And of course, Jerry Gleich, eosinophils are pathogenic effector cells in allergic diseases. Now, the role of the eosinophil in health and disease is an old question. Back in 1922, Huber and Kessler said that the tissue eosinophilia is a phenomenon far-reaching bearing, which it seems to us, if completely understood, would undoubtedly greatly elucidate the pathogenesis of asthma. In the 70s, eosinophils were thought to be good cells, into the 80s actually, 70s and 80s. But with the advent of, and they were thought to be good cells because of the concept that they released enzymes that would degrade mediators of anaphylaxis. This, of course, was shown to be untrue. And through the work of Jerry Gleich, exploring the functions of eosinophil granule cationic proteins, that changed from the eosinophil being a good cell to the eosinophil being a cell that played a role in disease pathogenesis. And of course, this was under debate for many, many years. And you can see Jerry Gleich here debating with a variety of luminaries in the field on whether the eosinophil is important in asthma and other allergic diseases. So this was the subject of many pro-con debates. And also, more recently, the roles of eosinophils in normal physiology, metabolic, and tissue homeostasis that I'll come back to. As a mentor, I'm showing a picture here from Eosinophils 1992. Jerry brought me as a postdoctoral fellow at this meeting in Adair Manor in Ireland, where I presented. And you can see many luminaries of the field here. You may recognize many of them. It was a very, very insightful meeting and set me off on my career. So as far as Jerry's role in mentorship, this was a very important point in moving my career forward. So the roles of the eosinophils in disease pathogenesis are now recognized. There are a range of diseases with prominent eosinophilic involvement. They're listed here. And those eosinophils have been shown in many, many studies to contribute to complications in a variety of gastrointestinal, cardiovascular, respiratory, hematologic, and cutaneous diseases, and also neurologically, and I'm sure Allison Fryer is going to talk about eosinophils and neurons in her presentation. Most notably, eosinophils play a prominent role in asthma pathology through the release of many, many mediators, including cytokines, the eosinophil granule cationic proteins, interleukin-5, and others, to impact the airway in terms of inducing airway hyperresponsiveness, mucus production, goblet cell metaplasia, tissue damage to the airway epithelium, and ultimately airway remodeling through the production of a variety of growth factors, not the least of which is TGF-beta. But in addition, eosinophils are becoming appreciated to have some roles in normal tissue and metabolic homeostasis and health. The major functions of the eosinophil, including their prominent role in remodeling and maintenance of tissue microenvironments, both during normal organismal development, but also as part of disease pathogenesis. And also to play a role in regulating post-innate and adaptive immune responses. But as we've found, in the absence of eosinophils and pathologies in eosinophil deficient humans, as noted by this editorial that Jerry and Chris Leiferman submitted to the Mayo Clinic, the question is, are they important in humans? And much of the data I'm showing here, much of the roles in normal metabolic and tissue homeostasis are findings from mouse that have yet to be confirmed in humans. So this is still a question up for grabs. Jerry's contributions to eosinophil science started with his role in describing the biochemistry, structure, function, molecular biology of eosinophil granule cationic proteins. He investigated eosinophil granules and discovered 3 novel proteins, MBP1 and 2, and eosinophil-derived neurotoxin. He localized MBP1 to the crystalloid core and showed that it was one of the most prevalent granule constituents on a molar basis. Before the Human Genome Project, Jerry's lab cloned genes, including MBP1, MBP2, EDN, and ECP, and characterized and identified pro-MBP, the pro form of MBP1. And they also identified a novel non-coding RNA called EGO that regulates the transcription of eosinophil granule cationic proteins. Jerry's lab was very prominent in studying the functions of these granule proteins in vitro and in vivo, and using purified granule cationic proteins. He demonstrated that they were markedly cationic. They functioned as toxins that can damage helminth, worms, bacteria, and mammalian cells. He identified the cationic ribonucleases, EDN, and ECP as cationic ribonucleases with enzymatic activity as potent as pancreatic ribonuclease. He demonstrated in collaboration with others that MBP1 damages bronchial epithelium, can cause bronchial narrowing, airway hyperreactivity when instilled into the bronchial tree of monkeys, and cough in the murine respiratory tract, and that MBP1 could stimulate histamine release from basophils and mast cells. So Jerry has contributed in a major way to our understanding of the conservation of form and function for the various granule cationic proteins shown here from vertebrate species and also over evolution. MBP1 action as a cytotoxin for damaging airway cells, parasitic helminths, is dependent, turns out, on its ability to bind heparin. And Jerry demonstrated with collaborators, including David Jacoby and Allison Fryer, who will speak today, that MBP1 inhibits lung smooth muscle M2 muscarinic receptors, causing constriction of the lumen. In addition, other parasites have proteoglycans on their cell surface, which were shown to be killed by MBP1, and lung epithelial and basal membranes also contain heparin-like proteoglycans on their surface. And membrane MBP was shown to cause the disruption of membrane potential in artificial bilayers and cause cell lysis. Ultimately, through Jerry's work, the structure of MBP, 3D structure, crystallographic structure, was identified, and it was shown to be a C-type lectin that binds heparin instead of mannose. Shown here, MBP1 shares overall topology with the C-type lectin family, but unlike the C-type lectins, which bind mannose, MBP1 binds heparin. And the cytotoxicity of MBP on epithelium is likely due to its ability to cross-link proteoglycans on cell surfaces, leading to disruption of transmembrane potentials. And Jerry has taken this further and is now developing radiologic imaging technique for detection, diagnosis, and monitoring of EOE based on heparin binding to extracellular MBP in the esophagus in eosinophilic esophagitis. Now, in terms of the history of Jerry's contributions to eosinophil research, I think there are a number of seminal papers that his group published while I was there as a postdoctoral fellow. And one of them is a paper using immunofluorescence microscopy that showed that MBP1 is deposited into the airways of patients with severe asthma at sites of tissue damage and mucus plugging. And this paper, I think, is a seminal paper that led to a paradigm shift in the field's understanding of the eosinophil's role as a pathogenic effector cell in asthma. And here's the paper in which, the Lancet paper, showing a few of the results here with MBP fluorescence in areas of mucus plugging and damage of the airway epithelium. And this paper, published in 1982, appears right here. And in my opinion, this was an inflection point. And I think this stimulated a huge amount and growth in eosinophil research over the following years. So I think this was a landmark paper that came out of Jerry's laboratory. In terms of the functions of the granulocationic proteins in disease pathology, Jerry and Chris Leiferman demonstrated that MBP1 coats the tissues of patients with numerous eosinophil-associated diseases, atopic dermatitis, the EGIDs, eosinophilic esophagitis in particular, and that the majority of eosinophils in those situations are activated and undergo a cytolytic degranulation with free granules and cationic proteins in the epithelium. Interestingly, in eosinophilic gastroenteritis, early on, this is in 1992, very early on in the EGIDS research, Jerry and colleagues demonstrated that MBP1 was present extracellularly in the absence of intact eosinophils in the gastric tissue as a biomarker for eosinophil activation, secretion, and cytolytic cell death in these GI and diseases and in other tissues. And so the eosinophil secretes these cationic proteins, and many of them have the ability to impact the production of remodeling factors. And since one of the major roles of the eosinophil is as a cell that's a remodeling cell that can induce pathologic fibrosis in eosinophil-associated diseases, these cationic proteins can induce the production of these remodeling factors. And here you see Jerry, he's been exposed to eosinophils perhaps too long, but unfortunately some of this is reversible. So you can see that he's now back to his normal self. So the eosinophil as a tissue remodeling cell and inducer of fibrosis produces many, many mediators that impact multiple aspects of tissue remodeling. And this is an example of tissue remodeling and fibrosis in eosinophilic esophagitis. And Jerry has made major contributions through various work done in his laboratory and in collaboration with others to define many of these factors and how they may impact a variety of aspects of tissue remodeling and fibrosis and eosinophil-associated diseases. Now, eosinophils as pathologic effector cells in the early 20th century, most scientists believed that eosinophils dampened the anaphylactic reaction by degrading mediators of anaphylaxis, and which I mentioned earlier was shown to be incorrect. And Jerry and colleagues showed that abolishing early on, very early on, abolishing eosinophils in animal models, guinea pigs, for example, failed to alter the severity of anaphylaxis, but demonstrated that they had roles in anti-parasite immunity. Now, Jerry suggested that eosinophils' role in inflammatory diseases such as asthma and hypereosinophilic syndromes was therefore largely as a mediator of disease pathologies. And ultimately, antibodies to IL-5 were shown therapeutically to benefit patients with eosinophilic asthma, strengthening their function as mediators of tissue damage and inflammation. And treatment of eosinophilic patients with benralizumab, which completely abolishes eosinophils, shown to decrease exacerbations, steroid dependency, and improve airways function. But there were some bumps along the way, and this 2000 paper by Peter Barnes, Leckie and Peter Barnes and colleagues basically said the eosinophil must rest in peace, that they have nothing to do with asthma pathogenesis. And you can see Jerry's reaction to this up in the upper left-hand corner. But ultimately, the eosinophil won out, and we now appreciate the eosinophil's role and contributions to the pathogenesis of eosinophil-associated diseases, thanks to Jerry's persistence in terms of resurrecting the eosinophil and moving research on it along to the point that now we have biologics that can be used to effectively treat asthma. Jerry also made contributions to allergic diseases in general and described a number of novel clinical syndromes. The most famous, perhaps, is episodic angioedema with eosinophil, the Gleich syndrome, shown here, but also played a role in identifying eosinophil myalgia syndrome and a variety of other clinical syndromes. And you can see Jerry here doing some telehealth care during the COVID pandemic. Now, Jerry was awarded the Paul Ehrlich Lectureship, which is awarded the highest honor bestowed by the IES on an individual who's demonstrated outstanding, prolific, high quality, and broad research achievements that have improved the knowledge of molecular mechanisms governing eosinophil functions in health and disease. And you can see here Jerry receiving the Paul Ehrlich Lectureship Award from Hans-Uwe Simon at the 9th Biennial IES Symposium in 2015 in Chicago. Jerry also has a long history of service to and mentorship of the eosinophil research community and the IES. He served as IES president from 2005 to 2007 and organized the meeting that was the 5th biennial meeting shown here that was held in Snowbird, Utah, not far from Salt Lake City, where Jerry resides. And in terms of service, Jerry has funded the Gerald Gleich Award. Jerry and Chris both together have funded the Gerald Gleich Award, which was established in 2011, recognizing the most intriguing and high impact finding related to eosinophil biology that was published in 2 years prior to the IES Biennial Symposium at which it is awarded. And here I'll finish up. Thank you. And here's Jerry conjuring eosinophils, and I'll take any questions. Thank you.
Thank you very much, Steve, for that wonderful overview. I see no questions in the question and answer box, and it's probably a good thing so that we can advance in terms of the time. If we have time at the very end, I might eventually have a little question to put to you, but maybe it would be also a good question for Jerry himself. So now it's a pleasure to introduce our next speaker, Hirohito Kita, who is professor of medicine and immunology at the Mayo Clinic. Hirohito trained as a medical student in Japan and worked as a postgraduate at Mayo under Dr. Gleich's supervision. Since then, he has continued to study mechanisms of eosinophil activation and mediator release, and currently runs a laboratory at the Mayo Clinic focusing on immunological mechanisms of allergic diseases and identification of novel therapeutic strategies. Dr. Kita will be talking about eosinophil migration and activation in tissue today. Hirohito, thank you very much. You can share your slides now.
Okay, can you see my slide okay?
Yes.
Okay, thank you very much. I'd like to thank the organizer for kind of inviting me for this kind of special webinar. Today, I'd like to talk about the eosinophil activation with kind of specific focus on a very unique aspect of the eosinophil biology. As Steve mentioned, in the 1990s, we started to able to isolate human eosinophil from the peripheral blood. That was when I moved to Gerald Gleich's laboratory at Mayo Clinic in 1988 and have been studying the activation of the eosinophils for almost like 20 years. So this is a slide which Dr. Gleich showed to me very early in my career. This is a patient with asthma. You can see the desquamation of the epithelium, but also you appreciate the eosinophil accumulation within the airway lumen. And by the anti-MBP staining, I think we can recognize eosinophil infiltration, but as well as the degranulation as well. So question is, how are eosinophils get activated and release their granular proteins? And this was the question I was asked to address. This is a tissue of the liver, and this is a patient with infected with Schistosoma mansoni. You may see the very similar picture as a patient with asthma. Here is the granuloma, and you can see the degranulation of eosinophils in the center of this granuloma where the parasites are. You can also see the intact eosinophils just surrounding this granuloma as well. Dr. Gleich and his colleague made a model to for this parasite infection. They used cephalous bees as a parasite and coated them with immunoglobulins. As you can see, serum IgG is very good at to induce eosinophil degranulation, as we can see in the release of the EDN. But surprisingly, secretory IgA was much more potent than IgG. IgA by itself was modest effect. By this picture, I think you can appreciate how the cephalous beads coated with secretory IgA are kind of attacked by the eosinophils and almost like a grape-like structure eosinophils are attaching to these cephalous beads. We use the similar system and we compare, in this case, we use the plastic plate to coat to the plate with secretory IgA or serum IgA. In case of the eosinophils, in response to the secretory IgA, they are highly activated and they produce superoxide. as compared to the serum IgA, secretory IgA is much more stronger. In case of the neutrophils, secretory IgA and serum IgA is not much different. Maybe secretory IgA is weaker to stimulate to neutrophils. So eosinophils are very unique at their response to the secretory IgA. And the question is why? And we think one of the reasons is a secretory component. In this experiment, we have seen that the secretory component and GM-CSF synergistically induce activation of the superoxide production by the eosinophils, which is not happening in the neutrophils. Another unique feature of eosinophils is they like to stick. I think many of you might have recognized this. So in this case, we coated the human plate with the human IgG, and as you can see, eosinophils stuck very tightly to this IgG. But when we block beta-2 integrin by anti-CD18 antibody, this eosinophil adhesion was totally suppressed. Neutrophils also react to IgG, but their reaction was not inhibited by the anti-CD18 antibody. Superoxide production, other marker for the eosinophil activation, was also suppressed by the anti-CD18 antibodies. And in fact, we think that this cell adhesion is a secret to induce activation of the eosinophils. In this case, we stimulated with eosinophils PAF, another good stimuli for the eosinophils. And with adhesion, they continue to be activated and have a sustained activation as looking at the chemiluminescence responses. Without adhesion, this sustained activation of eosinophils are not observed. This is likely to be the outside-in signaling of the integrins, because activated integrin activates Src family kinase, which react with adapter proteins and or Fc gamma receptors, leading to the activation of the Syk and downstream pathways. And eosinophils are very good at using that system. But why eosinophils are so sticky? One of the reasons we think is tetraspanin, like a CD9 and CD81, which is expressed on the eosinophils, but not on the neutrophils. Tetraspanin does not have their own ligand, but they are very good at making a web or network with other molecules, such as G protein couple receptors, integrins, and other molecules. And in fact, we can see the localization of the CD9 on top of the pseudopods of the eosinophils. And this likely be the secret life of the eosinophils. Eosinophils can also react with the products of the microbes, in this case, fungus alternaria. By incubation with extract of the alternaria, we can see the degranulation of eosinophils. Looks like it is a compound degranulation of the eosinophils. Using CD63 expression as a marker for the eosinophil degranulation, we can clearly see eosinophils respond to alternaria extract and release granular proteins, probably better than PAF. However, in neutrophils, they do not respond to alternaria, although they respond well to the PAF. The result of this interaction with fungi is likely to be the killing of the fungus. Without eosinophils, this alternaria is very happy. But when we add eosinophils on top of these alternaria, their membrane is compromised, and they become positive for the propylium iodide. Here in scanning EM, we can see this is a fungi, and I think we can appreciate how the eosinophils aggregate on top of these fungi. They release free granules, and I think you can appreciate like glue-like structures generated by the eosinophils, which is a mix of the DNA and granule proteins, and other molecules coming from the eosinophils. Why eosinophils are able to recognize fungi? And we think it is due to their ability to recognize the protease activities within the fungi. In fact, that activity of the extract to induce eosinophil degranulation is consistent with their protease activities, especially the asparatate protease activities. And when we use pepstatin to inhibit asparatate protease activities, their ability to induce eosinophil degranulation is totally lost. Eosinophils express PAR, which is a protease activated receptor, and proteases coming from the fungi cleave this N-terminus peptide from this receptor, changing the structure of this receptor and leading to the activation of the G protein coupled receptors and the downstream pathways. Finally, eosinophils can recognize other molecules such as ATP and uric acid in the tissue. In this case, eosinophils express highly P2Y2, which is a WAP purinergic receptor. They respond to the ATP and produce IL-6, which is inhibited by the anti-P2Y2 blocking antibodies. In response to the crystals of the uric acid, eosinophils produce IL-6 and IL-8, which is also suppressed by the anti-P2Y2 antibodies, suggesting that eosinophils has an autocrine ATP, which is released by the eosinophils and come back to the eosinophils to stimulate their purinergic receptors. So in a summary, eosinophils have a different way to activate. When get activated, we didn't talk about resting eosinophils, but they, in response to the cytokines and some of the soluble molecules, such as ATP, they produce cytokines and growth factors likely important for the immune regulation and tissue repair function of eosinophils. When eosinophils react with immobilized IgG immunoglobulins, such as secretory IgA or product of the microbes, they are completely activated, leading to the degranulation of the eosinophils, tissue damage, and killing of the microbes. But as a result, eosinophils die. Interestingly, this full activation of eosinophils does not seem to be happening in the mice. So this here, we compare the human eosinophils and mouse eosinophils using a variety of the stimulation. Human eosinophils produce superoxide in response to these agonists, but mouse eosinophils were able to induce this activation of the eosinophils, although they react with chemicals such as PMA. We think one of the reasons of this mouse eosinophils is lack of the several genes, which is important for the activation of eosinophils. Mouse genomes does not have a Fc gamma R2A, C, or Fc alpha receptor, or CD66 family molecules, which is GP-anchored proteins, and important for the activation of the leukocyte. This is simply missing in the mouse genome. So mouse models may underestimate the role of human eosinophils in the disease. So what triggers eosinophil activation? Probably many immunoglobulins, cytokines we didn't talk about, IL-33, adhesion through the beta-2 integrins, and activity and structure of the microbes and tissue microenvironment. I'd like to conclude that human eosinophils has a variety of unique tools to interact with the molecules in the mucosal tissues. I would like to acknowledge Dr. Gleich for his mentorship. This is the first paper I published with Gerald, and we studied the effect of the dexamethasone on the eosinophil degranulation. As you can see, this was a totally negative result. We didn't find any inhibitory effect of the dexamethasone. It took about 2 years to publish this paper, but I learned a lot. about the excitement of the research, rigor, and reproducibility on the experiment. In fact, if without any negative control, probably we have concluded, we have led to the wrong conclusion in this study as well. Trust between the mentor and mentees, optimism, resilience, and important for the support for the junior investigators. I learned all from this paper and subsequent interaction with Jerry. I acknowledge Jerry as a life mentor for me, but also acknowledge many of the students and fellows who contributed to this project and grants from the NIH. Thank you very much. I'll stop here. Thank you.
Thank you very much, Hirohito, for providing us all this insight on the diverse factors that can trigger eosinophils. We have one question in the question and answer box. Could you comment on the potential risk of giving anti-IL-5 therapy to patients who have presence of fungal allergic fungosis, and so with presence of alternaria or aspergillosis in the airways?
Yeah, that's a very good question. I think by the in vitro experiment, clearly eosinophils are ability to kill the fungi. But in vivo and in our body, probably we have a several redundant system which are able to deal with fungal infection using the other cell types and molecular pathways. So probably without eosinophils, I think we are okay with the resistance to the fungal infection. That's kind of my my speculation. Good question. Thank you.
Thank you. And since we have a couple minutes, I don't know if there are any other questions, but perhaps while we wait to see if there are any other ones, I do have one question for you regarding when you look at the strength of the secretory component in triggering eosinophils. I was just wondering if there's any relevance in human disease. Has there ever been any demonstration, for example, at mucosal surfaces and chronic rhinosinusitis, that IgA at that surface is triggering eosinophils?
Yeah, I think that's a good component. Clearly, secretory IgA is released into the lumen of the airways of the GI tract. And how they contribute to the activation of the degranulation of the eosinophils. Clearly, we see the degranulation of eosinophils in the very mucosal mucus of the patient with asthma and eosinophilic diseases. So maybe secretory IgA maybe contribute to this activity of the activation of eosinophils in these mucosal organs.
Okay, thank you very much. There's one question which has appeared in the meantime. Do you think anti-IL-5 therapy might impede B cell function in some scenarios since they express the receptor as well?
About IgA, say that again, sorry.
So I
Yeah,
think, so
yeah.
I'm just, yeah, so I think that this question is about anti-IL-5 therapy impeding B cell functions, which also express the receptor. Maybe this is referring to the IL-5 receptor in the question about it being present
Right.
in humans versus mice, or
Yes, right. That's right. I
translating
think
correctly?
clearly the B cells have IL-5 receptor, and IgA production is dependent on the IL-5 in the mice. But I think it could be the differences between the mouse and kind of humans.
Okay, and we have one last question. So would patients with secretory IgA deficiency who get recurrent airway infections, would eosinophil activation be less in these conditions? What are your thoughts about that?
No, I don't have any report that IgA deficient individuals and role of eosinophils in these lack of the resistance to the infection. I don't see any kind of report regarding the connection with IgA. Clearly, the IgA is important. How important eosinophils are for that? I think I don't have any answer for that. Thank
Okay,
you.
thank you very much, Hirohito. So we have one last question, but I think that it might be interesting to save it for Jerry's talk. So if we can leave that in the question and answer box, that would be great. Okay, so Let's move on to the next talk. I'm really very honored to introduce Allison Fryer, who is currently president-elect of our society and associate dean for graduate studies in the School of Medicine at Oregon Health and Science University. She's a longstanding and committed IES member, having organized a remarkable symposium in Portland, Oregon in 2019, just before the pandemic. Her lab is well known for its pioneering work on eosinophil nerve interactions with important implications in asthma pathogenesis, amongst others. Her work has helped lay the foundations of the now well-accepted concept that neuroimmune interactions contribute to chronic inflammatory diseases. This is what she will be covering today in the next talk. Allison, the floor is yours.
Okay, great. Thank you. So thank you so much for inviting me to talk today. I've been collaborating with Jerry Gleich, along with my husband, for over 35 years, and I'm really excited to share some of the science that we've been working on today. So the title of the talk is The Physiologic Interaction of Eosinophils with Nerves. And before I get into the eosinophils, I thought I would at least cover something about the role of nerves in asthma. So what you're looking at is a three-dimensional image of a whole mouse lung. The trachea is in blue at the top, and you can see all those beautiful branches into the bronchi and bronchioles. And what I really want to draw your attention to are to all those little fine white lines that you see down around the bronchioles. And here's a slightly larger picture of those. Those are all airway nerves. And the lungs contain an enormous collection of airway nerves. And it's very hard to appreciate those if you've been studying lung physiology using histologic sections. You really need to look in a three-dimensional analysis. And so this is another three-dimensional picture. And by the way, both of these images were generated by Greg Scott, who is a graduate student in our lab and is currently faculty now at OHSU. What you're looking at here is the inside of a bronchi, and then you can see at the bottom right, a branch into a bronchiole, and everything green is a nerve. These are mixed populations. There are sensory nerves as well as parasympathetic nerves in this image. And you can see all those little grape-like structures. Those are the parasympathetic ganglia. So the reason I wanted to show these is that the nerves are actually control bronchoconstriction. And we know this from multiple different animal models as well as humans with asthma. So what you're looking at here is a study from a dog, and they made the airways radio opaque and took x-rays of a dog while they were breathing. And you can see on the far left that this is the control phase and the airways are quite wide open. In the middle, they've exposed this dog to antigen. And so you can see, right where I've put those red circles, that those airways have constricted and have even closed, some of them. If they then treat the dog with a drug, atropine, that blocks all of the neural control of these lungs on the far right, you can see that these airways reopen. So this shows in dogs that in antigen-challenged dogs, that bronchoconstriction is actually mediated by the parasympathetic nervous system. The same is true in mice, and this is this is a mouse model of antigen challenge. In a control animal, if you just focus on the left side to start with, increasing doses of serotonin or 5-HT cause a very tiny bronchoconstriction, and those are the open circles labeled control. In antigen-challenged mice, this bronchoconstriction is enormously potentiated, and those are the purple closed circles. But if you cut the vagus nerves in the mouse, you can see that antigen-induced airway hyperactivity is completely abolished. So in mice and dogs, also in guinea pigs, if you cut the vagus nerves, you can completely remove airway hyperactivity in antigen-challenged animals. The same is also true in humans with asthma. Anticholinergic drugs cause bronchodilation. So this is a cartoon of this neural reflex, and this was provided by Ali Pincus in her PhD thesis. And what she's done is shown the trachea in the back, and then you sort of imagine a bronchi coming towards you, and it's cut in cross-section, and you can see that the bronchi is lined with epithelial cells, those little cubes with the circles in them. There are two nervous systems that are really important in the control of the airways. The first are sensory nerves, and these have their nerve endings in the airway epithelium, and those are those little black squiggly things. And they generate a reflex. So in response to any irritant or chemical stimuli that you inhale, that will activate those sensory nerves, and they will stimulate a reflex that's mediated through the brain stem. And then that triggers the parasympathetic nerves shown in red to fire. Now, the parasympathetic nerves actually provide the dominant autonomic control of the lungs. They're firing all of the time, and they release acetylcholine onto M3 muscarinic receptors that are on airway smooth muscle. And that causes airway tone. Even in a normal human being, if you inhale a muscarinic antagonist and block those M3 receptors, there will be some bronchodilation. So that's how you maintain normal tone. But when the sensory nerves fire, that reflex will then increase the firing of the parasympathetic nerves, increase the release of acetylcholine, and increase bronchoconstriction. Now, the release of acetylcholine also has a local control mechanism. And so these are M2 muscarinic receptors, different from the M3 on airway smooth muscle. The M2 are on the nerves themselves, and they inhibit the release of acetylcholine. So they function as a negative feedback mechanism and normally limit the release of acetylcholine. So in this image, I've got M3 receptors on the airway smooth muscle, and then the M2 are on the airway nerves. And these receptors, the M2 receptors, were discovered by myself and Dr. McLagan in the 1980s. So now how does all of this tie in with asthma and eosinophils? Before I get there, I do want to tell you how we measure physiology. And so most of the experiments I'm going to show you today were conducted in guinea pigs and in mice. We have also confirmed this in rats. The method is the same. The animals are anesthetized, and we cannulate the trachea to both ventilate the animal and also to measure inflation pressure. Both nerves, both vagus nerves on the left and right sides, are cut, and they're placed on stimulating electrodes. If you stimulate the vagus nerves, then you can get bronchoconstriction, and that's seen as an increase in inflation pressure, and that's on the far right. You can see that funny little peak. Stimulating the vagus nerves at the diamond and the VS causes bronchoconstriction. And so this is one of the ways that we actually measure M2 muscarinic receptor function. If you just look at the top trace for a few moments, you can see as we stimulate the vagus nerves, you get a bronchoconstriction, and that's shown as that peak that goes up and comes down. To study M2 muscarinic receptor function, we use a specific muscarinic agonist that stimulates those M2. Those M2 then inhibit the release of acetylcholine. And so there's a steady decrease in the bronchoconstriction in the presence of a muscarinic agonist. And that's what you're seeing at the top. So increasing doses of an agonist by activating that M2 receptor decreases acetylcholine release and decreases bronchoconstriction. The bottom trace is taken from an animal that was antigen challenged. And here you can see that that muscarinic agonist has no effect on vagally induced bronchoconstriction, meaning that those M2 muscarinic receptors are no longer responding to a muscarinic agonist. If you lose M2 muscarinic receptor function, we've measured that you increase acetylcholine release more than 10-fold, and you can increase bronchoconstriction 10-fold. And that looks a little bit like this. So in an antigen-challenged animal, we have shown that the M2 muscarinic receptors are no longer responding to inhibit acetylcholine release. There's more acetylcholine, and then the physiologic bronchoconstriction is shown on the right. You can see that we increase nerve stimulation in a control animal. We get a frequency-related bronchoconstriction, which is the green, and that this is significantly potentiated in antigen-challenged animals, and that would be the blue. So what is causing loss of M2 muscarinic receptor function? And it turns out that it's eosinophils. You wouldn't be surprised by that. I was invited to talk today. So eosinophils mediate loss of M2 muscarinic receptor function, and they mediate airway hyperactivity. If we get rid of eosinophils with an antibody to interleukin-5, we can protect the M2 muscarinic receptors on the left side of the screen, and we can prevent airway hyperreactivity, and that's shown on the right side of the screen. So eosinophils are tied up with M2 muscarinic receptors and airway hyperactivity in antigen-challenged animals. So how are eosinophils doing this? And the first question we asked then is, where are the eosinophils? And we talked about this with Jerry Gleich as well. And he relayed a story to us about telling a widow that her husband had just died, and she collapsed from an acute asthma attack and actually died herself. And so when we said to him that we think the airway nerves are very much involved in asthma and that eosinophils are involved with the airway nerves, his response was, well, that's of course they are. Let's figure this out. So in collaboration with Jerry Gleich, Richard Costello started to look at where eosinophils were in the tissue sections. And he started in guinea pigs, and this is the, Richard Costello's work. On the left is a tissue section from a control guinea pig. You can see the airway epithelium on the left. There's a band of smooth muscle marked SM. But underneath all of that, there are nerves and blood vessels. And I've marked a nerve with an arrow and an N. So that is a nerve bundle running through the lungs. The middle section shows a tissue section from an animal that's been antigen challenged. In this section, the epithelium is in the bottom left. There's a band of smooth muscle, which actually looks quite normal. But now draw your attention to that nerve bundle with that arrow in the N. And the last picture is of a close-up view of that particular nerve bundle. And you can see it is absolutely surrounded by eosinophils. And the arrow that has little asterisk in it is showing an eosinophil that is inside of the myelin sheath. So eosinophils are around the airway nerves. Richard counted the eosinophils and showed that there were more eosinophils associated with nerves than there were anywhere else in the lungs, except the airway epithelium. So there were more eosinophils around the nerves than there were in the airway smooth muscle or around blood vessels or randomly distributed through the tissue of the lungs. There were more around the nerves. He also showed that the same is true in humans who have died from fatal asthma, and these slides are provided by Jerry Gleich. And so what you're looking at is tissue sections from humans. The eosinophils are stained red with an antibody to major basic protein, and the nerves are stained black with an antibody to PGP 9.5. And you can see, if starting at the bottom left, that the nerve bundles are surrounded by eosinophils. On the bottom right, those are parasympathetic ganglia, and there are two eosinophils tucked right into the middle of those ganglion cells. And then at the very top, those are the fine nerve fibers that actually run through the airway smooth muscle. And you can see eosinophils are lined up along these nerve fibers. And once again, Richard showed that there were more eosinophils around airway nerves than there were anywhere else in the lungs. I also want to draw your attention to this little area here on the right-hand side. This is major basic protein that is not associated with an eosinophil, and I'll come back to eosinophil or major basic protein deposited in the lungs in a few minutes. So this is a movie showing of a human biopsy from a human asthmatic. The nerves are stained in green. Nerves with substance P are yellow, but the eosinophils are pink. And I think you can see this kind of three-dimensional analysis allows us to really carefully measure now where eosinophils are in relation to the nerves. And you can see this movie will stop looking at this eosinophil, which is really right on top of a parasympathetic nerve ending. So how are the eosinophils getting to the nerves? So one of the questions we asked is, if there's more around the nerves, what is going on in the nerves to actually recruit eosinophils? And it turns out that human and guinea pig parasympathetic nerves express eotaxin. They also make ICAM. And if you look at the top right-hand side, those are parasympathetic nerves grown in culture, and you can see eosinophils are adhering to those nerves, and we can interfere with eosinophil adhesion to nerves by blocking the antibodies to eotaxin, with antibodies to block the eotaxin receptors and antibodies to block ICAM. Okay, so nerves are actively recruiting eosinophils. So how then does that eosinophil change the function of the M2 muscarinic receptor? So it turns out that we've discovered the first endogenous antagonist. Eosinophil major basic protein will reversibly bind to M2 muscarinic receptors. And that is work done here. So Jerry Gleich provided us with a beautiful vial of human eosinophil major basic protein. And we tested it on receptors, muscarinic receptors obtained from guinea pig heart, which is the M2 muscarinic receptor. And we obtained M3 muscarinic receptors from guinea pig parotid glands. The receptors were all labeled with N-methylscopolamine, which will bind all muscarinic receptors. And then we displaced the N-methylscopolamine from the receptors using major basic protein. And you can see here that major basic protein in a dose-related manner will displace NMS from the M2 muscarinic receptor, but will not displace from the M3, showing that eosinophil major basic protein is a selective endogenous antagonist for M2 muscarinic receptors. The KD is 1.4 times 10 to the minus 5, which many of you might think is quite high, except that I'll show you later on that major basic protein physiologically is glomming on to the nerves throughout the lungs, suggesting that this is a physiologically relevant effect. We also showed that EPO is a weak M2 antagonist, but so weak that we were not able to calculate a KD. Eosinophil cationic protein and neurotoxin had no measurable binding to M2 or to M3 muscarinic receptors. I'm not showing it, but this binding is also reversible with heparin, as described by Dr. Keaton a few minutes ago. So it's all about MBP. And what we then shown in vivo is that if we block or remove major basic protein, we can protect the M2 receptor or prevent and reverse airway hyperreactivity. So remember, I talked about non-cell associated major basic protein, and that's shown here. On the left, you can see in human fatal asthma, everything that is red is major basic protein, everything black is a nerve. And at the very top, that nerve actually sort of, you can see it making an inverse Z shape, and it is shooting off to the left. You can't even see the nerve. It is so covered in major basic protein unless you focus the microscope up and down. And the same is true in a guinea pig. There's very little major basic protein in a guinea pig in the control, but following antigen challenge, you can see, and that's all the red. Major basic protein is everywhere, and that little green structure in the middle, those are the parasympathetic nerves. So major basic protein is present all throughout the lungs and specifically around the nerves in humans who've died from fatal asthma and in antigen challenged guinea pigs. So about this time, Jerry Gleich called us up and said, you know, I was cleaning out our -80 freezer, and I found many vials of an antibody to guinea pig eosinophil major basic protein. And he says, do you think you could use it? So we said, yes, of course we can. And he sent it to us, and we treated our guinea pigs with his antibody. And you can see that if we pre-treat animals with an antibody to major basic protein, we can completely prevent airway hyperreactivity in sensitized animals. And you can see that that's down below in the red. I'm not showing this data, but there are still eosinophils all throughout the lungs in these animals. I'm also not going to show you the data on the M2 receptor. Take my word for it, though, that the M2 receptor is also protected if we remove eosinophil major basic protein. We also wanted to test whether major basic protein was irreversibly damaging those M2 receptors, or was it really functioning as an antagonist? And it turns out that we can use heparin to remove major basic protein in vivo. So what you're looking at, and really just focus at the top peaks here. This is a trace from an antigen challenged guinea pig, and we're stimulating the vagus nerves at 1-minute intervals. And you can see that we can get reproducible bronchoconstrictions, those first 3 to the left. If we then give heparin and continue stimulating the nerves, over the next 20 minutes, that vaguely induced bronchoconstriction is reduced, and you can see that those peaks are getting smaller. If we test the function of the M2 muscarinic receptors, we know they're dysfunctional in antigen-challenged animals, but 20 minutes after heparin, their function has been completely restored. So eosinophil major basic protein is a reversible endogenous antagonist for M2 muscarinic receptors. So eosinophils and parasympathetic nerves are still the only mechanism to explain increased bronchoconstriction in antigen-challenged animals and people. So in summary, for this part of the talk, in sensitized animals, eosinophils are actively recruited to airway nerves, where they release major basic protein, they block neuronal M2 muscarinic receptors, increase acetylcholine release, and cause airway hyperreactivity. And if we deplete eosinophils or prevent their recruitment to the airway nerves, if we block eosinophil major basic protein, all of these things will protect the M2 receptors and prevent airway hyperreactivity. And you can see all the very different pharmacological interventions that we have tested. So I now want to conclude by talking very quickly about the sensory nerves. So these, remember, were the nerves in the airway epithelium. And this, we think, is yet another mechanism of eosinophil-mediated airway hyperreactivity. First, eosinophils can stimulate sensory neuron outgrowth. This is work done by Erin Foster in our laboratory, and she took sensory nerves and grew them in cell culture. And in the presence of eosinophils, you can see they put out many, many more axons and neurites, and we do not know the chemical mediator for this effect. But in vivo, we see very much the same thing. Eosinophils increase sensory nerve density in the airway epithelium, and that is linked to airway hyperactivity in mice. This is work done by Greg Scott. And if you focus just on the green to start with, Wild-type animals have eosinophils, and we've measured nerve density in the airway epithelium. This is significantly increased in the IL-5 transgenic animals, which have a lot of eosinophils in their lungs, and that would be the red bar. So you can see a significant increase in nerve density, and that is in parallel with a significant increase in bronchoconstriction on the right-hand side, comparing the wild type to the red transgenics. And if we get rid of eosinophils in the Phil mice, you can see that that limits the nerve density and it prevents this increase in airway hyperactivity. And the same is true in human asthma. This is work done by Matt Drake. He's measured in biopsies from patients with asthma that sensory nerves are increased in their airways and that is correlated with eosinophils. And so the data I'm showing you here, if you look at the green lines, those are maps of the airway epithelial nerves. And you can see that there are significantly more nerves in the airways of patients with severe persistent asthma, and that he's quantified that on the right-hand side in the red bar. And again, significant increase in airway nerve density, and that this is correlated with eosinophils. So in conclusion, eosinophils cause airway hyperactivity by increasing sensory nerve density and also by increasing the acetylcholine release from the parasympathetic nerves. As always, I need to thank a great many people, and some of their names are listed here. But I also really want to thank Jerry Gleich. We've had 35 years of collaborating, and this has been over dinner with antibodies, with slides, with conversations, long phone calls, talking about our families, our science, and philosophy. And I'm so grateful for our friendship. So thank you so very much.
Thank you very much, Allison. For the sake of time, I'm going to ask one very rapid question because it came up very early and it's pretty similar to the second one that came up. How does the relationship between eosinophils and nerves compare to that with mast cells and nerves? And a brief answer if possible.
That's a really good question because mast cells are one of the first inflammatory cells associated with nerves. We have never seen an effect of mast cells in our studies, but Brendan Canning and Brad Undum have done a lot of work on that and have shown that there are mast cells around parasympathetic ganglia and that they seem to be physiologically involved in increasing neurotransmission through those parasympathetic ganglia. And so that may be yet another mechanism for increasing parasympathetic control of the airways.
Thank you, Allison.
Thank you.
Okay, so this is the moment we've all been waiting for. Dr. Gleich's projections for the future of research on our beloved cell. I think I've said enough about Jerry and impatient to hear his talk that will be spirited and enlightening as always, despite his very recent and perhaps still ongoing struggle against a malevolent virus that has so unexpectedly changed our lives in many ways. But there have been some nice things associated with that, like the creation of the IES webinar series, and we're lucky enough, thanks to that, to be able to hear Jerry today. Jerry, thank you so much for being with us. We don't need to wear masks to listen real close. We're all ears.
First of all, thank you for allowing me to be here, Mr. Webb. Without your kind cooperation, this would be very difficult. First of all, what are you seeing on your screen now?
We see your slides. It's perfect.
Say again? Oh, okay. Okay. So before I start, I want to thank all of you for this great honor, and especially to Steve, Hirohito, and Allison for their remarks. I should also say that I've had the great pleasure to work with my wife, Chris Leiferman, for the extent of our marriage, and in 2 years we'll be celebrating 50 years of marriage and 50 years of talking about eosinophils. For example, last night, Chris says, "You've got to see this picture." And she's showing me a picture from her current work. But one more point. One thinks that people in science get excited by the results. And I have to say, the joy of science is in the doing, is in the interacting, is in the working. The results, the papers, the conclusions, they're great fun, of course. But I think we underestimate the pleasures of commingling with colleagues, of working, of simply sharing the joy of pursuing what we hope to be is truth, especially in these troubled times. So with that aside, my task is to talk about the future of eosinophil research. So disclosures should be on the screen, and the main disclosure I have is that I and my colleagues have started up a commercial firm called Nexios Bios, and I'm the chief scientific officer. I will show some data that that preceded the company. So if you're going to talk about the future, it's useful to look at the past. And to look at the past, we might have to go back to Paul Ehrlich. Ehrlich was really a remarkable guy. He identified a way to stain blood cells and thereby established hematology. He identified all the cells important for allergy and therefore established the basis of that. He established that B cells were important and how they were important, and thus established a fundamental feature of immunology. And last, but surely not least, Paul Ehrlich established the first pharmaceutical discovery laboratory for intelligent drug discovery, the first one ever. And many of you have heard of arsphenamine, for example. So after Ehrlich did his work, means for enumerating eosinophils were established, both in blood and in tissues, and shortly thereafter a relationship to anaphylaxis was discovered. And Steve Ackerman's gone over this very nicely. And that concept that the eosinophil was an important cell in controlling anaphylaxis lasted until the very recent times, surely as late as 1980. And around that time, there was a patent produced which actually claimed that eosinophils should be drawn into the lung to control asthma, something that we don't think much of now. Now, once the granule proteins were discovered and it was shown that the granule proteins were potent toxins, then that was a new concept, that the eosinophil could be an effector cell causing damage. And the next, I think, conceptual advance was the Lyre hypothesis. I'm sad to say that Jamie Lee, a close friend and collaborator, is no longer with us, but Jamie and his colleagues established or proposed that the eosinophil was a regulator of local immunity, and the abbreviations are listed there. So how about now? What's our present understanding? Well, it's clear that eosinophils are a mediator of disease, and this has been already touched upon. Benralizumab is a particularly important antibody. What role do eosinophils have in normal health? Back to Jamie and the Lyre hypothesis. Well, there are a couple of examples. There are patients reported in the literature without eosinophils. These patients often have other diseases, so it's difficult to say that they're not perfectly normal people, but they don't have any distinctive constellation of symptoms. There is no syndrome, evidently, that identifies them as being without eosinophils. And in fact, one of my patients in my current medical practice, by chance, I happened to notice that she didn't have any eosinophils, followed that up. There are no eosinophils in the bone marrow, and yet she's a diabetic, she has chronic urticaria, she's surely not a healthy, normal person, but again, no particular clinical syndrome that you can put your finger on. But perhaps benralizumab is the most telling experiment ongoing. Now, a while ago, there was a paper published in the Mayo Clinic Proceedings, actually, and Chris Leiferman and I submitted a response criticizing the paper because of the results with benralizumab. The authors responded saying, well, yes, benralizumab 30 milligrams every 8 weeks probably is not the most effective way to get rid of eosinophils. Well, that's going to change because benralizumab has now been approved by the FDA for the treatment of eosinophilic granulomatosis with polyangiitis. And the dose there is 30 milligrams every 4 weeks. And that's pretty effective in getting rid of eosinophils. So it's going to be most interesting to see whether any particular distinctive features emerge in those treated patients. As yet, there's no real data to suggest that that'll occur. I would like to talk about a little bit about pregnancy, and if Amy Cleon, if you're on here, forgive me, and you can correct me later, perhaps if I get it wrong. But Amy did an experiment testing benralizumab for the treatment of HES, hypereosinophilic syndrome. Beautiful paper published in the New England Journal. One of her patients was a woman who was treated with benralizumab and really did very well. So well that after the double blind, the open label, the patient was continued on benralizumab. And Amy told me, and I hope I get this right, that the patient came back on one occasion during the open label, and after doing the evaluations, they had her sit down, named Mrs. Jones, Mrs. Smith, whatever it was. We have to tell you, you're pregnant. And the patient broke down in tears because she'd been attempting to become pregnant, had not been successful. Well, benralizumab was continued because the patient's disease was so well controlled. And the patient went on to term, delivered a healthy baby girl. The baby girl had no eosinophils until about 8 weeks postpartum, when eosinophils became detectable and measurable in the blood. So what this experiment appears to tell us is that eosinophils are not critical for the most important feature of human existence, namely the ability to generate new people. There's Dr. Cleon's report is in the literature, and I think there is yet another report so stating. So what about the roles of eosinophils in normal health? Well, eosinophil subsets are being investigated. It'd be interesting to see whether we can develop good markers for those so as to follow their activities. There's recent papers suggesting eosinophils may have an important role in controlling colon cancer. That seems to be a target. In our work, we have focused on eosinophil degranulation, and a recent paper from the Fahy group in San Francisco investigated the measurement of the eosinophil peroxidase, EPX, in the sputum of patients with asthma. And in so doing, they showed that that was a very useful marker. I think Steve Ackerman contributed to the discussion of that paper. We also have continued to do experiments with immunofluorescence, and my close colleague, Dr. Leiferman, and has continued to work on this. The next slide showed you in the upper left a stain of an eosinophil biopsy from a patient with eosinophilic esophagitis, and the biopsy is stained with antibody to MBP. And you can see, I'm hoping you can see the green fluorescence, and you can see dots, which likely are intact eosinophils, but you can see hazy regions of immunofluorescence, which presumably represent MBP outside of the eosinophil. On the bottom in red is the computer's ability to measure the total tissue area, that's shown in frame A. The computer then is able to quantify that degree of fluorescence and relate it to the magnitude of the tissue area. And when that's done, frame B.
Jerry, we've lost your slides. We see you again.
Oh,
Can you share them again, please?
Let me share again. I'll try that.
I can see them.
Oh,
I
yeah.
can see them. Yeah, I can.
Okay, that's what counts.
We're
don't know what
back. These slides have a logic of their own. It's almost kind of scary. But suffice to say, when the computer quantifies the degree of MBP deposition, it says that 7.73% of the area is occupied by immunofluorescence, both cells and extracellular MBP. It then quantifies, Kristen has trained it so it quantifies the actual round eosinophils. That's about 1.36% of the total MBP. So by subtraction, 6.37% of the immunofluorescence is extracellular MBP. And Kristen has done these experiments many times. And we kind of summarized our findings in a publication. The true extent of eosinophil involvement of disease is unrecognized. It's unrecognized because pathologists don't see under light microscopy stained with hematoxylin and eosin, don't see the extracellular MBP. The secret life of dead eosinophils. That was Kristen's edition, kind of wonky. Okay. I'm going to try to move ahead here. I'll be jiggered. I have to say, guys, that my slides are posing a real challenge. Now they've taken off and just gone to town themselves. And don't ask me. I'll try to get back on. Okay. There we go. So another way to determine degranulation is radiological. And this shows an example of a patient with eosinophilic esophagitis who swallowed radiolabeled heparin. On the left is the SPECT image, single photon imaging computed tomography. And you can see the radioactivity in a long tube coming down. You can see the 3 blue fiducial markers. You can see the stomach. When you fuse the SPECT with the CT, you now can see the esophagus and the posterior mediastinum. And over on the right, you can see the volume of interest. So with this procedure, we can identify whether eosinophil degranulation occurs in the esophagus. We can see where it occurs, and we can quantify the degree to which it occurs. So this mimics the kind of data that one gets when one uses a gamma counter to measure radioactivity. We're in the midst of clinical studies here. I won't go further because these observations form the basis for the company that we started. Other questions. What's the mechanism of tissue dysfunction? In one of Steve's slides, he showed multiple possibilities. I don't think we know which ones are which. Next, could the eosinophil, as a bundle of cationic toxins, be trained to focus on cancer cells? Could the eosinophil be trained to be a cancer treatment? That seems like a possibility, but I'm not aware that it's been pursued. And lastly, we still don't know too much about the roles of eosinophil in normal health, and the new observations will be important. And last but not least, there are many eosinophil-related diseases which are listed here, and there's many episodes, many examples here to be pursued. And thank you for your attention. I'm happy to chat, take questions.
Thank you very much, Jerry, for your talk. The time is up, but I would just like to ask one question before ending this webinar regarding the extracellular deposition of granule proteins. What is known about the kinetics, the duration which they persist in tissue, the duration which they retain their activity? Could you comment on that?
Of course, yes. Well, first of all, we know that the injection of eosinophil granule proteins into skin of a guinea pig, admittedly, indicates that they persist for lengthy times, a couple of weeks with EDN and ECP, but MBP persists for up to 6 weeks detectable. We also know from a series of observations that the concentrations that one can observe by immunofluorescence are about 10 to the -5 molar. We don't know, however, whether these proteins retain activity during that period of time.
Thank you very much. And then I'm going to put that question to you, which came up earlier on. We would really appreciate your thoughts on, are there any potential drugs that could specifically inhibit degranulation of the eosinophil granule proteins, release of those proteins specifically?
Well, the degranulation, as Hirohito pointed out, is stimulated in a variety of different ways. I'm not aware.
Okay, thank you very much. Thank you for a wonderful webinar. I'd like to thank all the presenters for their outstanding presentations, and also thank the attendees for their interest in our society and for attending this webinar. The webinar recording will be available on our website and also on demand. And I encourage you all to please respond to our post-webinar survey so that we can further improve and better fulfill your needs and expectations. Have a nice day, everyone, or a nice evening for those who are joining from Europe or further. Bye-bye.